Bismillahirrahmanirrahim. Today we'll be talking about the avian embryology. Embryology of an organism consists of three main steps, that is, cleavage, blastulation, and now, first of all, let us know about the egg of the birds. Egg of the hen is megalacetal. Megalacetal means that it has a lot of yolk in it. The fertilization in case of the bird is internal. Fertilization of the chick egg occurs in the oviduct before the albumin and the shell are secreted upon it. The egg is telolacetal with a small disc of cytoplasm sitting atop a large yolk. The yolk egg of birds undergo discoidal meroblastic cleavage. Now, discoidal meroblastic cleavage actually means that the cleavage is restricted to only the small disc of the cleavage. Cleavage occurs only in the blaster disc, a small disc of cytoplasm 2 to 3 in diameter at the animal pole of the egg cell. The first cleavage furrow appears centrally in the blaster disc, and other cleavages follow to create a single layered blastoderm. These cleavages do not extend into the yolk cytoplasm, so the early cleavage cells are continuous with each other and with the yolk at their bases. Thereafter, equatorial and vertical cleavages divide the blastoderm into a tissue 5 to 6 cell layers thick. These cells become linked together by tight junctions. Between the blastoderm and the yolk is a space called the subgerminal cavity. This space is created when the blastoderm cells absorb fluid from the albumin, that is, the egg white, and secrete it between themselves and the yolk. At this stage, the deep cells in the center of the blastoderm are shed and die, leaving behind a one-cell thick area pellucida. This part of the blastoderm forms most of the actual embryo. The peripheral ring of the blastoderm cells that have not shed their deep cells constitutes the area. Now, gastrulation of the avian embryo. By the time a hen has laid an egg, the blastoderm consists of some 20,000 cells. At this time, most of the cells of the area pellucida remain at the surface, forming the epiblast, while other area pellucida cells have delaminated and migrated individually into the subgerminal cavity to form the primary hypoblast. Shortly thereafter, a sheet of cells from the posterior margin of the blastoderm migrates anteriorly to join the primary hypoblast, thereby forming the secondary hypoblast. The two-layered blastoderm, epiblast and hypoblast, is joined together at the margin of the area opaca in the space between the layer forms a blastocele. The avian embryo forms entirely from the epiblast. The hypoblast does not contribute any cells to the developing. The primitive streak. Uh, The major structural characteristic of avian, reptilian, and mammalian gastrulation is the primitive streak. This streak is first visible as a thickening of the epiblast at the posterior region of the embryo. This thickening is caused by the ingression of endodermal precursors from the epiblast into the blastocele and by the migration of cells from the lateral region of the posterior epiblast towards the center. As these cells enter the primitive streak, the streak become elongated toward the future head. As cells converge to form the primitive streak, a depression forms within the streak. This depression is what we call primitive groove, and it serves as an opening through which migrating cells pass into the blastocele. At the anterior end of the primitive streak is a regional thickening of the cells called the primitive knot or Hansen's knot. The center of this node contains a funnel-shaped depression, sometimes called the primitive pit, through which cells can pass into the blastocele. Migration through the primitive streak and formation of endoderm and mesoderm. The first cells to migrate through Hansen's node are those destined to become the pharyngeal endoderm of the foregut. Once inside the blastocele, these endodermal cells migrate anteriorly and eventually displace the hypoblast cells, causing the hypoblast cells to be confined to a region in the anterior portion of the area pellucida. The next cells entering the blastocele through Hansen node also move anteriorly, but they do not move as far as ventrally as the presumptive foregut endodermal cells. Rather, they remain between the endoderm and the epiblast to form the head mesenchyma. Now, this is the diagram of the movement of the cell. You can look at the Hansen node, the primitive streak. The upper layer is the epiblast, while you can also observe the blastocele, the hypoblast, and even the movement of the migrating cells can observe. Meanwhile, cells continue migrating inwardly through the lateral portion of the primitive streak. As they enter the blastocele, these cells separate into two layers. The deep layer joins the hypoblast along its midline and displaces the hypoblast cells to the sides. These deep moving cells give rise to all the endodermal organs of the embryo as well as to most of the extraembryonic membranes. The second migrating layer spreads between this endoderm and the epiblast, forming a loose layer of cells. These middle layer cells generate the mesodermal portions of the embryo and extraembryonic membranes. Now this diagram is showing different stages of development of the birds. Epiboly of the ectoderm. While the primitive mesodermal and endodermal cells are moving inward, the ectodermal precursors proliferate. Moreover, the ectodermal cells migrate to surround the yolk by epiboly. Thus, as avian gastrulation draws to a close, the ectoderm has surrounded the yolk, the endoderm has replaced the hypoblast, and the mesoderm has positioned itself between these two regions.